Hello, I'm Dave Tabb. I am a bioinformaticist working in the field of proteomics. Today I would like to show you how we can seek out orthologs, which is to say, if we have a protein sequence in one species and we want to see what the corresponding protein sequence is in another species that preserves the same gene, that's an ortholog search. And we're going to use the diamond sequence aligner to do that. To start, however, I'm actually going to start with NCBI BLAST as implemented over at Uniprot. If you're sitting at the Uniprot website, you can find the access to the BLAST just underneath the Uniprot logo. So here we are at the BLAST search tool. I have a sequence that I want to find an example of others in other species. Specifically, it's going to be this one, HP0318. This is a heme oxygenase, a metal binding protein that we know about from Helicobacter pylori. Now this is a really well-studied organism, but there are other species in the same genus that we also want to look within. Those include Accenonichus, Mustele, Hepaticus, Felis, and Bizazzaronii. And just as a bonus, our research colleagues made use of pylori strain B8 in their experiments, and pylori B8 uses a different naming strategy for its proteins than does the canonical H. pylori. So we need to find in all 6 of those species which gene is most similar in its gene product to this protein sequence right here. So I'm going to start by grabbing all of that and copying it to the clipboard. Now I can pop back over here to BLAST and enter this sequence as the query that I'm going to use. I'm searching throughout BLAST, throughout Uniprot for this particular sequence. And I need to specify pretty closely which part of the protein knowledge base I want to use. I think the default setting is for Uniprot knowledge base reference proteomes plus SwissProt. And that would sound pretty good, except that I'm actually not interested in stuff like plant proteins and human proteins, just stuff within Helicobacter. And I also know that some of the Helicobacter species I'm interested in are not in reference proteomes. They haven't reached that level of annotation yet. So I'm going to specify that I want to match against only microbial proteomes for this search. So now I'm going to leave the rest of these things all set to their defaults, nothing too special there. This will probably end up selecting, say, Blossom 62 as a matrix. It doesn't much matter to me. So in this case, I'm simply going to click Run BLAST. I wanted to note that I have a fair number of different proteins that I want to look up. I've got HP 10, 11, 129, 243, 318. You can see the list goes on. And I don't want to search in just one of the species. I want to search in 6 different species. So if you think about this being about a dozen proteins and this being 6 databases, 6 times 12 is 72 searches, which means I'm kind of sensitive about how long it's going to take to run each of these blasts. You can see we've run more than 35 seconds with no answer. In fact, this might run as long as an hour based on my experience this morning trying to work through this demo. So while Uniprot BLAST is a great solution and works very well, its response time can frequently be too slow for me to get the answer I want. Similarly, I can pop over to the NCBI BLAST Plus implementation at the European Bioinformatics Institute. This is very similar in its implementation to the way that BLAST runs over at Uniprot. And again, I can specify which type of proteomic knowledge base I want to search. I might start with all of Uniprot knowledge base, because as I said, some of it is not in the SwissProt well-annotated side or even a reference proteome. And I can also specify that I'm only interested in looking in bacteria. But even better, I think I'm going to stick with complete microbial proteomes, because I know that each of my species has at least a complete proteome on offer in Uniprot. I keep scrolling down. I can see that here I don't have to give it a protein database. I could have given it a messenger RNA search instead. But I'm going to do a protein sequence database search using the Hugsy sequence that I've provided here. BLASTp is the correct program to use because it compares protein sequences to protein sequences directly. BLASTx affords you a bit more flexibility for doing this searching. And finally, you can come down here and click Submit. You'll note that if you have a very, very long search that you need to run, you might want to specify be notified by email so that that result becomes available to you. Also, I have to keep this web browser window open long term to be able to see the search result that pops out of it. Having the email means that you have a persistent link to revisit that search result over the next week or so. Okay, I'm now going to click Submit, and I see that NCBI is also going to queue up a job for analysis. In my attempts to use this morning, I had results in about 5 minutes from NCBI Blast Plus on European Bioinformatics Institute, but over on Uniprot, it took me almost an hour, as I said, to get a search result. Okay, so that's wonderful, but wouldn't it be nice if we could run this search on this computer? This is a really powerful thing because now I don't need to wait in the queue for all the other jobs that may be set up to run on Uniprot or on EBI. Think about the fact that bioinformatics classes sometimes assign that everyone click the Submit Job button at the same time so that the server is instantly hit with 20 jobs that are all identical to run. That's obviously not ideal for responsiveness, and I would like to just use my own computer to do this search. So I'm going to use Diamond. Diamond is not Blast. They are not identical. However, Diamond is very, very powerful at accelerating searches that can produce scores very much like those of Blast. I believe that the statistical models used for producing e-values are the same in both of them. However, the way in which the search is accelerated is slightly different. Okay, so Diamond is something that I can download right here. You can see that there are both Linux and Windows builds, which is to say executable software that you can execute right away. It's also a really easy program to install because basically all you do is just copy the Diamond executable to somewhere on your computer where you can see it. In this case, I've thrown it in the directory with all the other files. So these are my queries from HP 0010 down to HP 1432. But for this purpose of this video, I'm going to just focus on this one sequence Hugsy, and these are the 6 sequence databases I want to query. Now, I've made a very quick bit of code here in a batch file to show the essential command lines that we need to execute this software. And the steps are really pretty straightforward. First off, the Diamond search algorithm isn't going to search these FASTA sequence databases just as they are. Instead, it's going to compile an index to make it much, much faster for sniffing through all of these sequences. To give you some notion of what that involves, Blast's strategy for speeding search is to look for places where a sequence in a subsequence in the query of 3 consecutive amino acids, say FGQ or VDP, any place where 3 letters from this query sequence can match any protein sequence in all of Acetonichus, in all of Mustela, etc. So simply looking for every occurrence of those 3 letters against a FASTA file, a big text file, is not efficient, but we can, using makedb, create an index to these sequence databases that is far, far faster for us to work with. So I'm going to run Diamond, which is on my path because it's sitting right in this directory, and use the makedb program and specify that our input file is going to be first Acetonichus. I'm going to copy that to the clipboard and just paste it like that. And I'm going to specify that the output from, I don't want to type uniprot proteome up blah 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 775 every time. That's much too slow for me. I'm going to call this ASIN, just the first 4 letters of the species name. So Diamond makedb, start with this FASTA file and create an output database called ASIN. Now that's extremely quick. Yeah, this is a good computer, but it's not that good. The fact is, bioinformaticists have done more to tune sequence homology search than practically any other bioinformatics algorithm. And so in this case, in just the space of a second or so, the software is able to read that all of the proteins from Acetonichus, I believe it's around 2,600 of them, find every occurrence of every possible 3-letter triplet in those sequences, and write that all out to a database file search, just like that. Oh, I see that the actual processing time was not 1 second, it was 1/25th of a second right there under total time. So you can see that we're really, really good now at making these homology searches, making the indexes for them very, very quickly. Now, I don't want to do just a Synonychus. I have a whole bunch of sequence databases I need to process here. So I'm going to run the diamond makedb on each of those in succession. All right, we already did a Synonychus. And now I need to give this one a name. I'm going to call this one must. Very quickly, we're through with that. Now I want to do one for hepaticus. I'm going to call it hep because I like to think of a hep cat. All right. And now I want to get my list of names back up here. Okay, bring my command prompt back. Now I want to do, let me see, we just did hepaticus. I need to do pylori B8, which remember is a somewhat different strain, and so it has different accession numbers. Oop, I don't want to call it hepaticus. I want to call that one pylori. Well, I'll call it pile. Okay. Now I need to do the felis. Okay, I'm just going to call that fel. I have one more to do. Oh, Bizazzaronii. Okay, so I'm going to click in that name. I'm going to write this one out as biz, which is fun to say. All right, so I started with a directory of just FASTAs and the diamond executable, really. Now I have these 6 additional files. I'm going to put those together by themselves. Okay, so we have asyn, biz, fel, hep, must, and pile. And I want to look for the occurrence of my particular sequence, this HP318, within that. All right, so we've already done the indexing. What's our next step? I'm going to type out my little example bat to remind me. So now that we've run makedb on each of the 6 uniprot reference proteomes or complete proteomes, I now need to run diamond in its blastp mode. You'll remember that blastp was something to compare protein sequences to protein sequences. When you run diamond in its blastp mode, you are telling it to emulate the behavior of blastp. I need to specify the name of one of these diamond databases. So I'm no longer telling it to use the uniprot FASTA itself. Now I'm using the diamond protein itself. Well, I don't even have to include the extension because bioinformatics people can be a little lazy sometimes. All right, and now I need to specify the Hugsy protein that I want to search. So where before we specified that makedb was to start with a uniprot FASTA and write out a diamond database, that had nothing to do with the query. So we only need to create this index once, no matter how many proteins we're looking for within that proteome. The blastp, on the other hand, it requires that we've already run makedb to create this asyn database, the DMND file. And we also note, by the way, that the asyn DMND, this index that we've created for asyn, is actually really, really similar in size to the uniprot that it started with. So it's a very efficient, very compact index that Diamond creates. Okay, so the Diamond blastp mode requires that we specify a Diamond index we're searching against and a query sequence that we're starting with. And rather than include that whole sequence on the command line, I'm going to specify that it's located in this FASTA database. So you might ask, how long is it going to take now to look for every sequence that has great similarity to this individual protein when it's looking at 2,500 or 2,600 sequences for a Synonychus? The answer is almost no time at all. I can pop back over here and I see that NCBI BLAST over on European Bioinformatics Institute is still running on this search. The BLAST search on uniprot is still running on this search. And something that this should tell you is that those servers are busy. They are serving a lot of people. So I can simply run this search on my own computer about as quickly as I can type the command line. Now, what we see in what returns from the blastp mode is a little complex, and I want to try to walk you through that. We start with the fact that it starts with Hugsy helpy. This is the query sequence that we provided in in HP0318. So we already knew that was our query, but the software reminds us this is what you looked for. And over on the right, it tells us this is the protein that we found that represented a high-quality match to this Hugsy protein. So Q17X53 underscore Halach. Okay, so one of the things we pick up from this is that acetinitrous is going under the name Hella. So Helicobacter, the first three letters of the Swiss prot identifier, of its suffix, is the first three letters of the accession of this trailer are showing what genus this comes from, and AH is the code for acetinitrous within that. But what about all these other numbers? Now, I might be able to piece out together that, okay, having a big number here is good, having a low number here is good, and that this is maybe how many amino acids are identical, but I'd rather not guess about that. And helpfully, if you pop over to the tutorial on the website for Diamond, you can see that each of these fields is reported explicitly. It defines what each of those means. So the things that we're going to care about, I think, are the sequence identity, which is to say what fraction of the amino acids were identical between the two. So if I flip back to this, I see that my number 93.2 tells me that 93.2% of all the letters were identical between my query sequence and the one that it found in the database. The 250s and so on, I'm not going to care about very, very much, but I will point you to the E value and the bit score, these last two values. So the bit score is a scoring matrix independent measure of the similarity of the two. Higher is better. So you can think of this as kind of the number of points that this scored, this particular homology did. And E value represents a way to say, if you were to run this search in random sequences, how likely are you to produce a value lower than this by random chance? So you want the E value to be nice and low. By default, you can see that Diamond is going to require that these are 1 part in 1,000 or better. So very, very low numbers are required for this to be a high homology. Okay, over here we see that the number of points scored for this sequence was 481, and the E value that results from that is 1.31 times 10 to the negative 176th power. This is a very, very, very strong homology found between a protein found in pylori and a protein found in acetinitrous. But that probably isn't true for all of these, and I'm going to go ahead and perform the searches now in the other 5 species. I'm going to start with Bizazzaronii, because it's next in alphabetical order. I'm going to continue to Felis. I'm going to continue on to Hepaticus. I'm going to continue to Mustele. And finally, I'm going to do the special strain of pylori that my collaborators used. Okay, so we now have, in every case, searched with Hugsy Helpy, and over at the right we have the best ortholog for it in acetinitrous, the best found for it in Bizazzaronii, the best in Felis, the best in Hepaticus, the best in Mustele, and the best in pylori. What would it mean if Diamond returned nothing? Is it possible that I could look for a sequence and not match anything at all in the sequence database? Of course. It could mean, in this case, that there is no ortholog for this gene, that gene might have been trimmed out by evolution's pressure to keep the microbial genome nice and small. So it is plausible that I would get no hit at all and no response from Diamond blastp mode. It would still pause, but there would be no response, no line of output produced. It is also possible that one of these species had a gene duplication, and so I might have more than one answer come back saying there are multiple significant hits in this FASTA database for this query sequence. Well, I think we can see that some of these sequences are extremely similar. Here we see that this last one scored 501 points and had a E value of 10 to the minus 184th power, and others produced slightly less good matches. Here we scored 239 points. Thought about on another level, you could say that in strain B8, we had a 98% identity between the two sequences, meaning that the amino acids were absolutely identical at 98.4% of the positions. But we can see that in some of these matches, we had far worse matching percentages. Here we had only 52.6% of the amino acids being identical. And it's likely that you want to see just how well those match against each other, not in a numerical score, but in a sequence alignment, to give you a sense of just how strong the match is for the amino acid sequences of your query and the amino acid sequence of what you found. Now, that's not output right here. It's a pretty compact output, in fact. So I'm going to need to find this particular sequence and then show it in a sequence alignment. The sequence alignment can be carried out over on the water server at MBOSS. Again, we are at European Bioinformatics Institute, and we see that we can do pairwise sequence alignment using the Smith-Waterman algorithm. Smith-Waterman is one of those algorithms that is has stood the test of time in a remarkable fashion. It is really astonishing when any bioinformatics algorithm outlasts the decade in which it was written, and the idea that it has outlived the century in which it was written is also very praiseworthy. Let us now enter two protein sequences that we want to compare to do an alignment, one against the other together. So I'm going to start by giving it the sequence of our query, which remember was found over in HP0318 FASTA. So I'm going to just type that to the command line. Now I'll grab that sequence and simply paste it into the web browser. Now I also need to get the second sequence, and that's going to be a little trickier. Remember, the one that we are getting is the one that had the lowest identity, and it was found in Hepaticus. So I need to look for this particular string in my Hepaticus database. Happily, I already have a text editor open on that directory, and so I can simply open the Hepaticus FASTA directory, FASTA file right there, and I can run a quick search for that string, and immediately I get an answer. It's this particular one. So I'm going to grab this thing out of here. This is just the same as doing a Control F, a big find in any text editor. The fact that I'm using Emacs is just because I'm basically very old. So let us now return to our Smith-Waterman algorithm, and I'm going to now copy this in as our second sequence. So just as a glance, we can look at this and say, this sequence starts MLNRIIE. This one doesn't start with that. It starts with MNHRIIE. So already we see some differences have crept in, even without having to do the alignment. But let's now proceed with that. I'm simply going to click the Submit button. We don't really need to do anything special to make this alignment look just right. Okay, now earlier we started an NCBI BLAST job. It's still running. We started an NCBI BLAST running over on Uniprot. It's still running. Are we going to have to wait as long on the alignment? No, we are not. This database search is, there's no database to search in this case. We simply have to align two sequences on each other, which means that it runs very, very quickly. I'm just going to click on the View Alignment File option because that's probably the best way to work. Now, I'm going to try to help you decode this because I realize it might not seem like this is a particularly strong match. I chose it because it was the weakest of all the strong matches that we had for this Hugsy protein. When you look at the symbols that appear between these two sequences, you'll see that some of them have just a space in between them. So here this is actually a gap. We have a gap that we've inserted in our query sequence to account for these letters that were added in the sequence that we found. So this is obviously no match between the two. That's why these are all spaces lying in between them. But down here, let me see, I saw an example of this just a moment ago. Oh, I can't find it now. But sometimes we have a weak amount of homology between two letters. Here we have an alanine, and here we have a glycine. You can, for the intensive evolution's purpose, you can think of alanine as a glycine with a methyl stuck on it. So we can understand why those are at least tolerable in matching to each other. Some of these are a little stronger, though. Alanine and serine tend to be a little stronger in their association with each other over evolutionary time. S and N, more similar to each other. Glutamine and glutamate, relatively similar to each other. Of course, isoleucine and leucine are a pretty strong match for each other. Those are all given double dots. And then we see all these vertical pipe symbols, and those represent places where the amino acids are absolutely identical. L is L, R is R, E is E, F is F. So when we look at these alignments, we're seeing that some parts of this sequence are very strongly retained, such as this stretch, where every single amino acid is perfectly identical between the two. And then you have regions like this gap, where there's absolutely no homology between the two. So you might ask which parts of this sequence are most conserved under evolutionary pressures. And that's where you find these long runs of identical amino acids. Okay, well, I hope that gives you a bit of a starting place to using Diamond. You can find it over at GitHub with this URL. I'll try to include that in the description for this video as well. And of course, it's available for both Linux and for Windows. So very handy software, very, very high performance. And if you're ever stuck in the place where you're waiting on the web server to do a job that you could do on your own machine much more quickly, I hope you'll think of Diamond, which is very, very powerful software for that purpose. And I hope that you'll take a look at some of the other videos that we've posted in bioinformatics education, and that you'll like them a lot. And if you see a subject that's missing, I hope you'll drop me a line. Okay, thank you very much, and I'll talk to you again soon.